Hello students, I am Akshay Deshpande and welcome to a biology class, and as you all know that I am your biology tutor. So today we will go with the further part of the same chapter, that is cell, the unit of life. Right, so today we are having the third class of the same chapter, that is the cell unit. And if you try to recall whatever we have discussed till now at this particular moment, we have mainly discussed about the prokaryotic cells, and we have also discussed about the overview of the cell. Right, so till now we have mainly discussed around 40 questions, right? And similarly we will also try to look on with some more questions in this particular part, and also we will try to discuss the further part of this particular chapter. Right, so without any further delay, let's start our discussion then. So we will start our discussion mainly on the eukaryotic cells, right? We are going to mainly start our discussion on the eukaryotic cells. Now, the term eukaryotic mainly means that they are having the true nucleus, right? True nucleus. Any organism that basically has the true nucleus, or any, we can say, cell with nucleus or nuclear membrane, right, with a true nuclear membrane, and inside this nuclear envelope or the membrane you mainly find the genetic material, that is the DNA. Right, so any cell that is having the true nucleus, or in other words a cell with the nuclear membrane, and inside this nuclear membrane if you find the DNA, and this DNA we all know it is mainly the genetic material. It is the genetic material, right? So then we call it as eukaryotic cells. Then we call it as what? Eukaryotic cells. Remember, there are unicellular eukaryotic cells, right? There are also unicellular eukaryotic cells, and similarly you also find multicellular eukaryotic cells also, right? For example, the protozoa. Okay, the protozoa are mainly the examples for the unicellular eukaryotic cells, and multicellular eukaryotic cells we are having many examples: plants, fungi, okay, etc. They belong to the multicellular eukaryotic cells, right? So now here we are having some examples: the protista and fungi, animals and plants, right? These are the most common and well-known eukaryotic living organisms. So now, one of the interesting feature of the eukaryotic cells is that you see extensive compartmentalization. You find extensive compartmentalization. Now, what do we mean by the term extensive compartmentalization? See, we can try to draw a diagram over here, and if you draw this as your animal cell, just for a representation purpose, you can see that this is how you will observe our a structure, that is the nuclear envelope, right? Yes, nuclear envelope, right? And here you find mainly the thread-like structure, that is the chromatin, right? Okay, and here you find concentric structures, right? Yes, so here we are having one more structure attached with this. Similarly, one more structure. What we are drawing over here, just a simple schematic representation, right? And here you find a tubule-like structure, and similarly dense dotted structures we can try to draw over here. And we can draw one simple structure, that is this we can say as our Golgi apparatus. And here we are having one structure called as the mitochondria, right? Yes. So here you can find vacuole like structures, right? So now here we can say that they have mentioned about the compartmentalization. Okay, compartmentalization in the sense making the formation of compartments. Okay, you find presence of compartments. Okay, and these compartments are very essential because they mainly form separations. Okay, they separate the cellular components. They mainly separate the cellular components, right? So for example, this part you are looking over here is mainly the endoplasmic reticulum, and this we call it as what? The Golgi apparatus, right? So now you can see that all of these are mainly dividing the cell into different compartments, maybe endoplasmic reticulum, maybe Golgi apparatus, right? Yes. Now similarly you are also having the nuclear envelope, right? So the nuclear envelope, what you're looking over here, is mainly responsible for separating the or isolating the DNA or the genetic material from the cytoplasm, right? So that is the mainly function of the nuclear envelope, right? So these membrane structures inside the eukaryotic cells, they are mainly playing an important role in the compartmentalization of the cytoplasm. They divide the cytoplasm into different series of compartments. So this is a unique feature of the eukaryotic cells. Now apart from that, you also find membrane bound organelles. You also find the membrane bound organelles. So there are multiple examples for the membrane bound organelles. For example, we are having endoplasmic reticulum, we are having Golgi apparatus or Golgi complex, and we are having mitochondria, we are having a chloroplast, and one more example we are having is the nuclear envelope, right? And we are having vacuoles, right? And we are having lysosomes, right, etc. Multiple membrane bound organelles you are mainly finding over here. Now you also find a nuclear membrane. You also find what? A nuclear membrane, and the main function of the nuclear membrane is to separate or isolate the DNA from the cytoplasm, okay, isolation of the DNA from the cytoplasm. That is the main function of the nuclear envelope, right? And also remember, the nuclear membrane is also a double membrane structure. It is also a double membrane structure. It is a double membrane structure. Now, not only you find these structures or compartments inside the eukaryotic cells, you find special structures called as the cytoskeletal structures. Okay, you also find the cytoskeletal structures. Now, what do you mean by the cytoskeletal structures? The cytoskeletal structures mainly include intermediate filaments, okay, yes, and one more we are having is the actin, okay, and one more we are having is the microtubules, right? All of these are actually the cellular skeleton. Okay, they mainly provide the structural support. Okay, they mainly play an important role for the structural support. Okay, see, the function of these cytoskeletal structures is very much more clear in the case of the animal cells. In the case of animal cells, the function is very much clear and evident, okay? Because in the case of animal cells, you do not find any rigid structure such as the cell wall, okay? But however, you find the structures In the case of plant cells, for example, you mainly find a structure similar to cell wall, right? The cell wall is mainly involved in giving shape and mechanical support to the cell structure. But in the case of animal cells, you do not find any such structure, right? So for the proper shape and motility, okay, structural support and motility, you find skeletal structures playing a very important role in the case of animal cells, right? So we can say that the function of the skeletal structures, cytoskeletal structures, are more pronounced or more evident, more clearly observed in the case of animal cells than anything else, right? And finally, inside the nucleus, you mainly find the chromosomes, which are mainly the DNA is compactly arranged, compactly formed or arranged, right? So DNA is compactly arranged to form the structure that called as the chromosomes. You will study about the chromosomes more in detail in your last part of this particular class. Right, so these are some of the important overview you find in the case of eukaryotic cells. So now, okay, we will erase this over here. Now we can look into some of the vital differences between the plant cells, and one more we are having is the animal cells. Okay, so this is one of the well-known question generally you will expect in your theory examination about mainly the difference between the plant cells and the animal cells, right? So what are the differences between the plant cells and the animal cells? See, the plant cells have an exclusive structure, that is the plastids. Okay, so plastids, we are having example that is mainly the chloroplast. Okay, the chloroplast is mainly observed in the case of plant cells, right? You find a rigid structure that is called as the cell wall, which is an exclusive character of the plant cells. You do not find such kind of structure in the case of animal cells. Instead, in the case of animal cells, you find in some cases, not in every organism, not in every animal cell, but in some cases, you find a proteinaceous sheath or a covering called as a pellicle. Okay, you mainly find a pellicle in the case of animal cells, but you don't find a rigid structure similar to cell wall in the case of animal cells. And you also see vacuoles. Okay, vacuoles are predominantly observed in the case of plant cells. And if we all know the diagram of plant cell, right? So if you draw a plant cell from your lower classes, this is how you have actually drawn your plant cell. And this is our mainly the cell, and inside the cell generally we draw a large empty cavity like structure. This is called as the vacuole, right? Yes, around 90% of the plant cells space is actually occupied by the vacuole itself, right? So it's a very big and dominant structure in the case of plant cells, but you don't observe such kind of vacuole-like structures in the case of animal cells, right? But animal cells are having a unique one more structure called as the centrioles. Okay, centrioles are quite unique structures that are mainly observed in the case of animal cells, but nowhere else. Okay, plant cells do not have centrioles, right? So what is the function of the centriole, if you ask? The main function of the centriole is mainly in the formation of spindle fibers. Okay, the main function of these centrioles is the formation of spindle fibers, and these spindle fibers are mainly helping in the cell division. We all know this, right? They are mainly involved in what? Cell division. Okay, how they are mainly playing an important role in cell division? That is separation of the daughter sister chromatids, okay, or daughter homologous chromosomes, right? So the separation of the sister chromatids is mainly because of pulling effect of the spindle fibers. As a result, those sister chromatids are separate, right? Hence they play a very vital role in the case of animal cells. Centriole plays a very vital role in the case of animal cells. But however, the centrioles are mainly absent in the case of the plant cells. Then obviously, then the most fundamental questions that you may expect is that if centrioles are absent, centrioles are absent in plant cells, right? Then how do the chromosome separate, right? How do the chromosome separate? This is a very interesting question that you people may ask, right? So the answer is quite complicated, and also it is quite high level to explain over here. Okay, here in the case of plant cells, generally this function, that is separation of chromosome or the sister chromatids in the chromosome, is mainly done by the motor proteins. Proteins. Now these motor proteins are mainly involved in the formation of spindle fibers. Okay, remember the spindle fiber formation, though it is not done by the centrioles in the case of plant cells, but that doesn't mean that it is not occurring. The spindle fibers still form in the case of plant cells, right? And these spindle fibers, which are mainly formed from the motor proteins, not from the centrioles. Okay, so those centrioles are not present in the case of plant cell. It's not a big deal. The suppression of sister chromatids and also the homologous chromosomes during the mitotic and meiotic events can still occur, and that is mainly done by the motor proteins in the case of plant cells, right? So these are some of the interesting aspects about the eukaryotic cells and some exclusive feature of the eukaryotic cells, right? So I hope I have made the points clear over here. So let us begin our discussion with the first important cellular structure, the most commonly heard structure in the case of both plant cells or animal cells, that is the cell membrane. Mainly what? The cell membrane. So now the structure of cell membrane was not known clearly. Okay, it was not known clearly. Okay, it was not known clearly, but however, with the help of the electron microscope, the detailed structures was mainly studied in 1950s. Okay, the structure was studied more in detail and elaborately in early 1950s and late 1950s, and this was mainly because of the electron microscope, the advent and invention of the electron microscope, and the cell model that was mainly used to study the cell membrane or the plasma membrane was mainly the RBC. Okay, so now the question is, why RBC was only used? Why RBC was mainly used? Okay, there are many cells inside the cell body. Okay, we are having WBC, we are having platelets, we are having any cell, right? But what made or what makes RBC quite special? Okay, so if you look into the cell structure of RBC, you know that it is completely anucleated. Anucleated. What do you mean by anucleated? It means that it doesn't have a nucleus. It doesn't have a nucleus. There is no nucleus, right? There is no nucleus. If there would have been nucleus, okay, if there would have been nucleus, what would have happened? There would be nuclear membrane also, right? So if we want to study about the cell membrane, we want only the cell membrane or we want only the plasma membrane. We don't want any other membrane structures from the cell, okay? But we also know that inside the cell there are also what? Membrane bound organelles, right? So it means that there may be every chance that during the study of a plasma membrane we may get one or two membrane structures apart from the plasma membrane also, right? So to avoid confusion or to avoid bias in our study, scientists mainly chose RBC because it is completely not having the nucleus, and even if it is having nucleus, it is only during the early developmental stages. As the RBC matures, you don't find the nucleus inside the mature or developed RBC. Okay, it is having only a protein structure, that is the hemoglobin. Okay, you find mainly the hemoglobin, HB. Okay, it is an abbreviation we use for the hemoglobin. Right, that is one particular point. And also you don't find any kind of membrane bound organelles inside the RBC. So when we are studying the membrane, we are studying only the plasma membrane. As a result, scientists mainly chose RBC to study the plasma membrane. Right, now during the study and analysis of the plasma membrane, scientists noticed that it is a chemically complex, and it is chemically mainly composed of two important molecules, that is lipids, and one more we are having is the proteins. Okay, it is mainly made up of two important components, that is the lipids, and one more we are having is the proteins. Right, so now in the case of lipids, you find an important lipid called as the phospholipid. It is called as what? Phospholipid. So now let us discuss about the phospholipid a bit. Okay, it is a important what we can say a constituent of the membrane to discuss. Right, so now the phospholipid is a very interesting molecule. Okay, why? Because the phospholipid has both the hydrophilic property, and also it is having the hydrophobic property. It is having both hydrophilic property and also one more property, that is the hydrophobic property. So any molecule, okay, any molecule that is having both the hydrophilic property and hydrophobic property, it is called as amphipathic. Okay, it is called as what? Amphipathic. And interestingly, the lipids in the membrane, that is cell membrane, that is the phospholipid, are the amphipathic in nature. Okay, so now let us look into the structure of the phospholipid. Generally you see that there is a polar head group, which we call it as the phosphate okay, phosphate head group. Right, and you find two tails. You find what? Two tails, and this tail is called as the fatty acyl tail, or sometimes they have also mentioned the what we can say the hydrocarbon tails. Hydrocarbon tail. Right, they have mentioned hydrocarbon fatty acyl chain or tail in your textbooks. Right, so let us go with that particular point only over here. So as we said just a moment ago, we mentioned that the phospholipid is what? Hydrophilic also, and also hydrophobic in nature. Okay, and you see that we have mentioned that the phospholipid is mainly made up of a phosphate head group and one more the fatty acyl tail. Okay, so now which part of the phospholipid is hydrophilic and which part of the phospholipid is hydrophobic? It is mainly the phosphate head group that is hydrophilic. So what do we mean by the term hydrophilic? It means that it is water loving, right? So it shows the affinity towards the water. This is what we mean by the term hydrophilic in nature, right? So now, but whereas the fatty acyl or hydrocarbon fatty acid chains are mainly the hydrophobic, right? They're mainly what? Hydrophobic in nature. So now, as they are hydrophobic, the term hydrophobic mainly means that they do not like water, they are water-fearing molecules, right? So, as a result, the lipid is actually the lipids in the case of a plasma membrane are mainly bilayer. They are mainly what? Bilayer. It means that it is mainly made up of two layer of lipids. They're mainly made up of what? Two layer of lipids. So this is quite an interesting point over here. Okay, so now if we have to draw this diagrammatically, we can draw the lipid bilayer in this particular manner. Okay, we can draw the diagram in this particular manner, right? And this is one layer of lipids. Okay, this is one layer of lipids, and similarly we can draw in this particular fashion. Okay, in this particular fashion. So this makes a second layer. So we can say that both these two layers, we can say it as what? Both these two layers as what? The lipid bilayer. Okay, so here we have mentioned that particular point, that the lipid arrangement is what? It is a bilayer, that is polar head group is towards the water and the hydrophobic tail is mainly interior, right? So you can see over here that this head group is exposed to water, but whereas the tail group are what? Interior. Okay, it is not exposed to the water group, it is buried inside. So now we have also mentioned that you also find proteins. Okay, you find also what? Proteins, right? The proteins are also present in the lipid bilayer, and proteins play a very important role in a functioning of the membrane. They play a very vital role in functioning of the cellular membrane. We will be looking into it shortly. So now the last point they have mentioned is the cholesterol is also present, right? Yeah. So now cholesterol plays a very, very important role in the cell membrane. Okay, so what is the function of cholesterol in the cell membrane? It mainly provides the fluidity. It mainly plays an important role in the fluidity, right? So let us erase this particular part. Okay, and we can draw the diagram once again. This is one phospholipid molecule, second, third, yeah, right? Yes. Similarly one more, similarly one more, and similarly one more, right? So now cholesterol, okay, cholesterol mainly plays an important role in the fluidity. Okay, what fluidity? Membrane fluidity. We mean by the term membrane fluidity. It plays an important role in the membrane fluidity. How? Okay, the membrane fluidity is mainly controlled by a factor, that is the temperature. Remember this. Okay, so now if the temperature increases, so fluidity increases, right? But however, if the temperature decreases, fluidity decreases. Remember, right? So if the fluidity is increasing, and one of the reason may be what? Increasing the temperature. If the fluidity is decreasing, then one of the reason is what? The temperature is what? Decreasing. Okay, yes. So these are some of the important points. So what happens is that as the temperature increases, the membrane becomes more fluid and fluid. Okay, what do you mean by more fluid and fluid? The distance between the two molecules, phospholipid molecule, increases. We can draw it over here. Okay, this will be one molecule, this will be one more molecule, this will be a third molecule, right? So the distance between the two molecules has increased, right? So this is called as what? More fluid. But however, if the distance between the two molecules is completely decreased, then we say that the fluidity has what? Obviously decreased. Okay, if the distance between the two phospholipid molecule is decreased, then we can say that the fluidity has decreased, but the distance between the two phospholipid molecule has increased. It means that the fluidity has also increased, right? So now if the fluidity increases, there will be more gap between the phospholipid molecules. In other words, there will be enough gap such that the molecules can enter inside the cell, right? So now this is an undesirable trait, or this is not something you want. As a result, what happens is that the cell starts to synthesize more and more cholesterol, such that the cholesterol molecule will start to sit inside in between the gap of the phospholipid molecules. Assume that this is a molecule of cholesterol, what I am drawing over here. Okay, so these molecules of cholesterol will sit between the distance or increased gap between the phospholipid molecules, right? As a result, we can say that the fluidity is brought back to normal, even though the temperature is increased, right? So now what will be the condition if the temperature has decreased? We said that fluidity has decreased because the distance between the two molecules has also decreased. So you can basically create the distance, the space between the two molecules by adding more cholesterol, So it is very much similar to the situation where the temperature has increased, right? So even in the case of temperature where it had increased, more cholesterol molecules were added. So similarly, even in the case of lower temperature, the cholesterol is again added to create some space between the phospholipid molecules. As a result, the fluidity is brought back to normal condition, right? So cholesterol is also present, and the main role of cholesterol is that it is mainly playing an important role in the fluidity. Remember this. This is a higher biology concept, but however, I thought of giving an explanation over here. Yeah, so this is mainly about the cell membrane and the arrangement of the cell membrane that we have discussed over here. So now you find that the proteins and carbohydrates are actually present. Okay, so carbohydrates, what do we mean by carbohydrates? We are mainly speaking of sugars. Okay, we are mainly speaking of what? Sugar molecules. See, I will give you one easy examples to understand the carbohydrate concept in the case of cell membrane. Okay, so what happens is that you remember the RBC, what we have discussed. The RBCs mainly contain the antigen. Okay, the blood groups, what you have studied, antigen A, antigen B, and also what you have studied for the blood groups, right? So the antigen is basically a protein molecule. It is basically what? A protein molecule, and this protein molecule is having a extra molecule attached to this, and this molecule is basically the sugar. Okay, so now what is the function of carbohydrate or sugar that is present in the cell membrane? They play a very important role in the cell recognition. They play a very important role in what? Cell recognition, cell interaction. They play a very important role in what? Cell interaction. Okay, so the carbohydrates are mainly playing an important role in cell recognition, cell interaction, and proteins are mainly involved in what? Adhesion. Okay, cell to cell contact, cell to cell adhesion is mainly done by the proteins. Remember this particular point. It's a function that we have to remember. Okay, so now let us look into the ratio of lipid to protein. Okay, generally it all depends upon the type of cell that actually we are studying. Okay, so if we are having human RBC, we can say that in the human RBC, 52% of it is actually protein in the lipid layer or the membrane, and 40% of it is what? Is mainly the lipids. Okay, 52% is the protein and 40% is mainly the lipids. So now proteins, we can categorize the proteins in the cell membrane into two types. The first one we are having is the integral proteins, and one more we are having is the peripheral proteins. So now what do you mean by integral proteins? Integral proteins are the ones that are completely buried in the lipid bilayer, right? They are mainly completely buried in the lipid bilayer, but peripheral proteins are mainly present externally. Okay, they are mainly present where? Externally. Okay, they are mainly attached to the lipids. They are mainly attached to where? Lipids, right? So now how do we classify the proteins? Okay, we are saying that it is integral protein. We are saying it is a peripheral protein. On what basis we are actually telling that the proteins in the membrane or the cell membrane is integral and protein peripheral? It is mainly based upon the extraction. Okay, so now what do you mean by extraction? Extraction means mainly removal. Okay, it mainly means what? Removal. It means that when the cell membrane is exposed to detergents, yes, you may not believe we are using the term detergents. Okay, detergent is a very simple term of explaining you, but we can say that membrane lysing agents, and generally the membrane lysing agents we use the term detergents. Okay, yeah, so don't get confused with the term of the cloth washing detergents. Okay, yes, we are mainly referring to the agents that are going to break the cell membrane. Okay, so membrane lysing agents, we also use the same term detergents for them also. Okay, so using these membrane lysing agents, we can basically bring some disruption in the membrane. We can disrupt the membrane. Okay, we can disrupt the membrane, and this disruption of a membrane can cause the removal of proteins, or we can say release of proteins, removal or release of proteins. So if the proteins can be easily removed, then we can say it is mainly the peripheral. Okay, peripheral. If they are buried in lipid by layer, then we use the term integral, or we can say that it is very difficult to remove, then we use the term integral. Right? So this is what we do the classification for the integral and peripheral proteins. I hope you have understood the concept behind the integral and peripheral proteins. So now, whatever model we have discussed, okay, this model is called as the fluid mosaic model, right? It is called as the fluid mosaic model. Fluid mosaic model. And this model was mainly proposed by Singer and Nicholson in 1972. Okay, see, there are many models that have been explained or tried to explain the structure of the cell membrane or plasma membrane, but however the most well accepted and the stapled theory in the cell biology that explains the plasma membrane structure is the fluid mosaic model, which is mainly proposed by the Singer and Nicholson, and the year in which they explained this was published was mainly in 1972. So now, according to this model, it says that the membrane is fluid-like. Okay, somewhat. The word somewhat is important. Okay, what do you mean by somewhat? The term quasi. Okay, quasi means similar to or somewhat. Okay, roughly, that is what we mean by the term quasi. Remember this. Okay, so the membrane is somewhat fluid like in nature, and the proteins are floating in the ocean of lipids. This is what they have described in this particular model. Okay, so Singer and Nicholson said that the membrane is somewhat fluid like. So that is what we mean by the quasi-lipid layer. Quasi-lipid layer. The membrane is what? It is a somewhat lipid fluid like, and the proteins are mainly floating in the oceans of lipids. This is what the original statement in their papers tells us. Okay, now because of this fluid property, okay, quasi-fluid or fluid-like property, we can see that there is a lateral movement of the proteins. There is what? Lateral movement of the proteins. So what do you mean by lateral movement of the proteins? Let us write a diagram over here. Right? So let us assume that there is also one more behind this, one more behind this, one more behind this. Yeah. So this is what a three-dimensional structure I am drawing over here. This is a three-dimensional structure, right? Yes. So on the surface of the lipid molecules, you can see that there is a protein molecule. Assume that this is a protein molecule. Okay. Yes. Now what kind of protein molecule I am drawing? If you look over here, the diagram what I am drawing over here is actually the extrinsic proteins, or also we can say that it is peripheral proteins, right? So in some cases the integral proteins is mainly what? Buried inside the lipid bilayer. Okay, it is mainly buried inside the lipid bilayer. Okay, in some cases the integral proteins, you may also find one more type that is called as the transmembrane. Transmembrane means completely passing through the double layer or lipid bilayer. It is called as the transmembrane. Okay, yeah. Anyway, so this peripheral proteins what I have drawn over here, this one, and this one. Okay, it can show lateral movement. Lateral means what? Sideways, in this way, or in this way, in this way, in this way. That is this way, or this way, or this way, or this way, right? It is called as the lateral movement of the proteins. So remember that. Okay, the lateral movements of the proteins is mainly due to the quasi-fluid like property or quasi-fluid like nature of the plasma membrane. Okay, so this movement is called as what? Fluidity. This movement we refer it as what? Fluidity. So we have made the concepts clear over here. I hope so. Now, the fluid nature is mainly required for the functions such as the cellular growth, intracellular junctions. Don't worry about this. We'll study in your structural organization chapter of animals. And the next one we are having secretions, that is the release of products. And one more we are having is the endocytosis. What do you mean by endocytosis? Exocytosis and endocytosis. For example, if this is the cell, a food molecule is entering inside the cell. Okay, is entering inside the cell, and the process of uptake of food molecules or any solid or liquid food molecules into the cell is called as the endocytosis. The last one we can use or we are even requiring this fluid nature of cell membrane for the cell divisions. So remember the functions, very important, that the fluid nature of the cell membrane is contributing to. Okay, yes. So please don't get it confused with the function of cell membrane. No, we are not discussing about the function of cell membrane. This is mainly the fluid nature of the cell membrane that is helping for all these processes. Okay, yes. The fluid nature of cell membrane is helping for all of these process, but not the cell membrane directly. Remember this. Okay, yeah. So let us look into the functions of the cell membrane. It is mainly involved in a transport of substances. It is involved in signaling, and it is involved in what? Adhesion. So now if it is involved in a transport of substances, for example, the osmosis, okay, passive diffusion, and also it is playing an important role for the active transport. Okay, you will study about these all mechanisms more detail in your the chapter that is the transport in plants. Okay, but in a nutshell we can say that active transport mainly requires energy, that is ATP. Now passive diffusion is just a movement of substances from a region of higher concentration to lower concentration, that is called as the passive diffusion, and it does not require ATP or energy. Osmosis is a movement of water molecule from a region of higher concentration to lower concentration. That is what we mean by the term osmosis. Now signaling we discussed. Okay, we will study about the signaling, that is a hormone signaling mechanism in the chemical coordination, right? Yeah, so for the signaling, extracellular receptors are basically present on the cell membrane only, right? Hence cell membrane is also playing an important role in the signaling process. Next is adhesion. Attachment of one cell with the another cell is mainly due to the cell membrane, because the cell membrane contains the important proteins that are involved in what? or involved in promoting the cell adhesion process, right? So these are few important functions. There are many important functions, but just the important ones that we have to discuss over here, right? Transport of substances, signaling mechanisms, and also it is involved in the adhesion. Now we can look into the plant cells. For the plant cells, they are having a different structure called as the cell wall, right? Now we all know the fungi and plants are also having this rigid structure, but there is a difference in when it comes to the chemical composition of the cell wall in the case of fungi and plants. Okay, we will be discussing that. But remember, in the case of fungi, it is mainly made up of chitin. Okay, so now the main function of the cell wall is mainly to promote the shape and give the protection and also prevent the entry of undesirable macromolecules inside the cell. Okay, it prevents movement of undesirable macromolecules inside the cell. That is mainly a function of cell wall. Okay, so now it mainly brings the cell to cell interaction, and also we have mentioned this prevents the undesirable molecules moving into the cell. We have discussed this, right? Yeah. Now if you look into the chemical composition, for example, the algal cell wall is mainly made up of cellulose, okay, galactans, mannans, and also minerals such as the calcium carbonate. But plant cell wall is mainly made up of cellulose, hemicellulose, proteins, and the pectins. Okay, so again, vital for your questions. Okay, vital for questions. Remember this. So here in the case of cell wall, you mainly find two types of cell wall, that is the primary cell wall and secondary cell wall. So secondary cell wall is mainly formed in the inner side of the plasma membrane, and it is mainly associated with the maturity. Okay, as the plant cell starts to grow, achieves maturation, then only you see the formation of secondary cell wall. But however, the primary cell wall is mainly observed in the case of young plant cell, and the primary cell wall is a flexible. Okay, it is what? A flexible, not a rigid structure. Okay, it is having a growth capacity, but however, it disappears or diminishes later stage. Okay, so after disappearance, then only you find mainly the secondary cell wall in the plant cells. So we are having the last concept over here, that is the middle lamellae. Okay, now middle lamellae is mainly a what we can say a glue-like structure. Glue-like structure, and this structure is mainly made up of the calcium pectinate. Okay, now in some cases, in some plants, okay, the neighboring cell cytoplasm may be connected directly. For example, if this is one cell, okay, and this is another cell, okay, the cytoplasm of both the cells are connected directly. Okay, so this connection we are mainly meaning by the term plasmodesmata, connects the cytoplasm of neighboring cells. Okay, so the molecules from A cell can directly move into the B cell, and B cell molecules can directly come into the A cell. Okay, that is the main function of plasmodesmata, right? So we have discussed the concepts quite elaborately over here. So what we can do is we can start to look into some important questions. We will erase this. Yeah, and the structure that essentially glues the neighboring cells together is the plasmodesmata, cell wall, plasma membrane, and middle lamellae. Just a moment ago we discussed that is the middle lamellae is mainly involved in gluing the neighboring cells together, right? It is mainly made up of calcium pectinate. Just remember this point. Okay, this is again a previously appeared question. This is a previously appeared question. Now, which of the following structure plays a role in connecting the cytoplasm of neighboring cells together? Okay, so that the molecules can move from one cell to the another cell. Plasmodesmata, tight junctions, middle lamellae, cell wall. We all know it is what? plasmodesmata. Okay, the two cells cytoplasm are connected directly so that the molecules can move from one cell to the another cell. Now, which of the following statement about the cell membrane is true? The membrane is made up of proteins only. Apart from lipids, cholesterol is also present. RBC, in RBC, the lipids are 52%. Proteins in the membrane is only peripheral. Okay, which of the following statement about the cell membrane is true, right? So, apart from lipids, cholesterol is also present. Okay, cholesterol is also present. Of course, even proteins are also present, but however, when we consider the lipid category, okay, when we consider a lipid category, apart from lipids, even cholesterol is also present. So cholesterol is the starting material for the synthesis of the lipids. Hence we can say that B is our right answer. Okay, so lipids are mainly 40%, the proteins are 52%, and protein membrane is having only peripheral protein. That is not true. It is having both integral and also peripheral proteins. Right? So now let us look into the next question. The lipids in the membrane are amphipathic, hydrophilic, hydrophobic, amphibolic. Okay, I have discussed this in the initial part. The answer is amphipathic. Okay, any molecule that is having both the hydrophilic property and hydrophobic property, it is called as amphipathic nature. Okay, and the phospholipids of the plasma membrane exhibit this property. Okay, yes. Now, the lipids in the membrane is arranged in bilayer, single layer, triple layer, quasi-layer. The answer is what? Bilayer. The membrane in the, lipids in the membrane are arranged in the bilayer nature. Okay, two layers. Now, which part of the phospholipid is exposed to water? Hydrocarbon chains, phosphate head group, proteins, all of the above. We know that the polar head group or also the phosphate head group. Polar head group, remember. Okay, don't get confused. All mean the same. Polar head group or phosphate head group are basically the what we can say phospholipid, which is mainly exposed to water. In other terms, extracellular matrix or the cytoplasm, both contain the water. Yeah, so the next question we are having is, which cells have been used to study the cell membrane structure initially? Okay, WC, platelets, basophils, RBC. We know it is what? RBC was mainly used to study the structure of cell membrane because they do not contain any other membrane bound structures inside the cells, right? It contains only the plasma membrane. As a result, we use the term RBC or the RBC cells. Now, the function of sugars in the cell membrane is cell-to-cell contact, linkage, interaction, none of the above. Okay, the main function of the sugars in the cell membrane is to provide interaction. Okay, interaction between the signaling molecules. Yes, important. Okay, though the carbohydrates or sugar are also involved in the cell-to-cell contact, but however the major role of the cell-to-cell contact is done by the proteins, remember. Okay, but interaction and making the cells aware about self and non-self is mainly done by the what we can say sugars or carbohydrates, right? So hence the right answer over here we are having is the interaction. C is our right answer. So next question, if the membrane proteins are complexly buried into the lipid bilayer, then such proteins are peripheral proteins, integral proteins, transmembrane proteins, lateral proteins. The answer we can say integral proteins. Okay, of course, though we are having transmembrane proteins, but remember transmembrane proteins mainly span or cross across the lipid bilayer. Okay, so the more appropriate answer for this particular question is what? B, that is integral proteins. They are complexly buried into the interior of the lipid bilayer. Yes, there are nothing called as lateral proteins. It doesn't exist. Peripheral proteins are mainly attached to the surface of the lipids. Remember this. Now, when the cell membrane is when the cell membrane lysing agents are applied, which of the following will be observed? Okay, what is the result they're asking, right? So the integral membrane proteins are released, phospholipids are disrupted, lateral movement of the proteins are observed, peripheral proteins are disrupted. Okay, so which is the easiest protein that can be removed from the cell membrane? It is the peripheral proteins, right? So if you apply the membrane lysing agents, the proteins that are affected first are mainly the peripheral proteins. Hence we can say that peripheral proteins are disrupted, right? So D is our right answer. So when peripheral proteins are disrupted, even the lateral movement of the proteins are also what? They are also disrupted. Remember, this is also another consequence of the using the membrane lysing proteins. Okay, so we are having the next question. Which of the following molecules are present in the membrane? Cholesterol, lipids, proteins, all of the above. The answer is simple. It is all of the above. All of the contents, all of the above mentioned are also present in the membranes. The next one. Sugar molecules in the plasma membrane are attached to transmembrane pumps, proteins, cholesterol, interior of the membrane. Okay, they are mainly attached to the proteins. Just go through the diagram of your what we can say in your textbook. You can see that it is mainly attached to the proteins. Okay, sugar molecules in the plasma membrane, they are mainly associated with the what we can say proteins, maybe integral or transmembrane or peripheral, any type of protein. Remember, it's just simply the proteins. Now, which of the following is an important function of plasma membrane? Okay, cell adhesion, transport of molecules, cell interaction, all of the above. Okay, so we can say that cell adhesion, transport of molecules, and also the cell interaction, all of the above. Okay, it plays an important role in all of the given above options. So D is the right answer over here. Polar molecules move through the plasma membrane by osmosis, pumps, carrier proteins, and one more we are having is the passive transport. It is mainly the carrier proteins. Okay, any examples we know for this? That is the glucose, right? Now glucose cannot cross the plasma membrane. Okay, so for the crossing of plasma membrane you find mainly transporter, okay, or we can even use the term carrier proteins, right? So carrier proteins or transporter molecules are mainly used for what? Transport of the polar molecules. Now let us look into the next question. Okay, the movement of water from the region of higher concentration to the region of lower concentration is mainly the passive diffusion, osmosis, active transport, facilitated diffusion. We all know the answer is what? Osmosis, the most simplest definition, and we have studied it multiple times. Okay, osmosis is your right option. The next one. Which of the following is energy dependent transport? Osmosis, passive transport, facilitated diffusion, active transport. The answer is D, active transport. It because mainly requires the ATP, and ATP is the energy that is required for transportation of molecules. So remember one important point over here. The transportation of substances or molecules in the active transport is against the concentration, right? So I did not mention this point. The active transport mainly transports the molecules against the concentration gradient. Okay, but whereas facilitated diffusion or passive transport or osmosis, they're all down the concentration gradient. Okay, as a result, they don't require energy. So let us remember this point. The next question. Okay, which of the following is in the cell wall of algae? Cellulose, glycans, pectins, proteins. Okay, so in the case of algae, it is made up of galactans, mannans, and cellulose. Okay, exactly, remember this particular point. And also calcium carbonate, let us not forget that. Okay, so cellulose is our right answer. So which of the following statement about the cell wall is not true? Okay, a false statement they're asking. The cell wall of plants is made up of cellulose, hemicellulose, pectins, and proteins. True. The primary cell wall has a growth capability. True. And secondary cell wall is added after maturity. True. Algae has minerals called as the calcium pectinate. No. Okay, it mainly has a calcium carbonate, not the calcium pectinate. Right? Remember this point. So as a result, D is a not a true statement or false statement, hence the right answer for this question. The next one, Na plus K plus ATPase pump is an example for active transport, osmosis, facilitated transport, and one more we are having D option, that is the passive transport. The answer is active transport. Okay, you will study about the sodium potassium pump more in detail in the nervous system chapter. Okay, neural control and integration, in that chapter we'll study about it, that is action, potential, conduction, and all. Okay, so I will not discuss about it detailedly over here. So we will look into one more question. Which of the following nature of cell membrane allows the lateral movement of the proteins? Okay, dynamic fluid property and quasi fluid property, solid property, all of the above. We know it is the quasi fluid property that mainly promotes the movement of molecules laterally, mainly the protein molecules laterally, right? Yeah, so we have mainly discussed around 20 questions in the part that we have discussed till now yet. Okay, mainly about the eukaryotes. Okay, so I urge you to solve more questions along with that what we have discussed over here. Try to go with some materials. Okay, any material that is available at your disposal, try to solve the questions from those materials. In this way you can be more proficient. Okay, so I will meet you students in another video where I will discuss the advanced part of this particular chapter. Thank you.